I have a PhD from the University of Illinois in Champaign-Urbana. I specialize in natural products and anti-infective agents. And after I got my PhD in Illinois, I postdoc at Harvard for about 2 years. I was an NIH postdoctoral fellow with Bob Woodward. And then after that, I started my career in American industry. Basically started it out at American Cyanamid Lederle Labs in Pearl River, New York, where I rose to the ranks, starting from individual contributor all the way up to worldwide director anti-infectives chemistry, and also chemistry support for molecular biology. I left there in about 1994. I went to the West Coast, and in the interim 20 some odd years, I have been involved with about 4 different startup companies. Several of them have gone public. Most are in anti-infectives related, but there's one of them is also in oncology, immunology related. Subsequently, I have been at a company here on the East Coast called Adhesives Incorporated. It used to be a privately held company where I was a CEO, CSO till roughly about late 2016 when we sold the company to Universal Display. I stayed on as the chief scientific officer and also advisor to the CEO and also to the Universal Display Ventures. Okay, I started as an individual contributor. I was put into the program, which is involved in the synthesis of new types of anti-cancer agents, things related to mitoxantrone and bisantrene. When I joined the program, bisantrene had been identified as a potential lead candidate for development. Unfortunately, at that point, there was an issue in regards to the CMC aspects of the molecule because its simplicity. And so basically, one of my jobs is to come up with alternative ways to generate the basic scaffold that's needed for bisantrene and provide opportunity to prepare other related analogs which are not accessible by the standard commercially available anthracene type molecules. And so that was basically how I got involved in the program at the beginning. Then later on, I got involved in the program making prodrugs because of the fact that I come up with a patent which allow various prodrugs to be made for long-term therapeutics and things like that. Well, it was one of several lead molecules they had. There was a compound called mitoxantrone, which we were also developing at that time. Mitoxantrone was further ahead of the game than bisantrene, but bisantrene had a differentiated anti-tumor activity, which basically they decided they wanted to explore further, which they did. They took it to phase 1 in France. It was an interesting environment. It was an environment where basically it was in a rebuilding phase, where basically in the late 60s they had lost a lot of world-renowned scientists. Many went to academia, and this is a situation where they trimmed down the headcount in order to protect profits due to patent erosion and things like that. And I was part of that group that was hired in the mid to late 70s to help to rebuild that organization scientifically. I think it's a combination of several things. Number one is that it was felt that its oncolytic profile could, if both drugs, mitoxantrone and bisantrene, were introduced on the marketplace, they could possibly cannibalize each other's market. Plus, on top of it, that mitoxantrone is further ahead in the development pathway and a little bit easier to synthesize than bisantrene itself. But the third item was probably because of the unusual solubility profile, which required them to use a central line catheter, which sort of detracted from its elegance. The material, bisantrene itself, has as a free base, is relatively insoluble in physiological pH. And it requires a little creative thinking about ways to try to change a formulation that is still going to be acceptable in the pharmacological world. And so basically, that's what I mean by elegance. It's one of these situations where it was a diamond in the rough. It's just a matter how you polish it up in order to make it clinically more accessible and useful for the clinicians. And I think the other thing is you have to look at this thing from a retrospective aspect, which is basically, in those days, drug formulations wasn't as advanced as it is now. And so, I find doing drug formulations very challenging, but it's not a challenge because it's really to understand physical organic chemistry of a molecule and its physical chemical property and just tweaking it and, thinking through the pathway. How you're going to administer it, where is the pH going to be in the pharmacological sense of the body, and you just work around it. This is not the first time I've had these issues in life. I've dealt with it a few times. I think they probably would have developed it. I really think they would have. This is one of those situations where organization under financial pressures makes a so-called rational decision and ends up putting a good drug up on the shelf and forgetting about it. And then patent life is such that it expires and then they just don't go back and revisit it. I think the other thing that people forget is that the treatment of cancer now is totally different than what it was 20, 25 years ago, where the thought process, you use one single agent and then you hope that that single agent carries with it. The thought process has changed significantly. In fact, what they do in cancer is just like they do in anti-infectives. Sometimes you use two different mechanisms of drugs at the same time in order to ensure a better positive outcome. And that's the same thing. They probably could have had that opportunity to use bisantrene as an adjunctive agent with another anti-cancer agent. You just don't know until you actually gone through a whole profiling process. Yeah, there's at least half a dozen that I can think of small molecules, and some of them would be like azacitidine, hydrochloride, which they had a very short half-life. They're able to make a longer half-life. Another one is cyclosporin, which many people are familiar with, called Neoro, which basically was a reformulation of a drug that had a low undesirable PKPD, which is used obviously for various types of immunological conditions. Then this mesalazine, which is they have now made a multi matrix release mechanism so that it's once a day type drug. Paroxetine, which is a Pfizer called Paxil CR, is extended release type formulations, things of that type. And then it's valproate, which many people who are epileptics use, has an issue with short half-life, which results in undesirable side effects. By changing the formulation, they spread out the half-life, but at the same time reduce the exposure rate to the point that you can take it like once a day and things of that type. Then there's also another one, viloxifene, which you call Effexor XR, which is not used as much, but again, it's an extended release type formulation. So those are some of the ones that I can think of offhand. Well, I think number one is always nice to have new opportunities in the oncologist handbook or armamentarium. That's one thing. Number two is that In contrast to, say, 20, 25 years ago, when pharmaceutical firms are looking for blockbuster drugs, the reality is when you are trying to create drugs for patient-specific conditions, this is a type of drug that we should be re-examined in its totality. And I really like to see that occur here because we have seen historically some drugs which appeared to be low financial potential turn out to be major contributors to some companies' bottom line because somebody was creative in thinking through the reformulation process.